Today, I will be mostly talking about contemporary evolution in arable weeds as revealed by the resurrection method. This is a project that I have started developing in 2020 when COVID started. And I finally have some interesting results and a lot of perspectives of what to do with these results. But I also am working on a lot of other things. So here is just a brief overview of all of the plants that I have been studying in the last 15 years. And these are all the places where I either did research or field work. So you can see that some of the plants are perennials, some of them are annuals, but the vast majority of them have pretty flowers. This is not only because it's pretty to look at, it's because I'm mostly interested into the evolution of plant mating systems in response to environmental variation. So a few short examples of what I have done so far before we dive into the resurrection topic. The variation of claystogamy in response to environmental cues, where I focused on the species Lamium amplexicoli that produces disclosed flowers, which are obligately self-pollinated because no pollen comes in or goes out. But as many claystogamous species, Lamium amplexicoli also produces open flowers that are potentially outcrossed. So I studied the variation in the production of open and closed flowers in response to a lot of environmental cues. And the latest paper being currently under revision examined this in response to interspecific competition, no competition, and the effect of drought stress in addition to the competition. Another project that was recently wrapped up, and we have a lot of papers lined for publication, but it's a big project with a lot of collaborators, so it takes time to get through the revision steps, was the citizen science campaign looking for cowslips. In Czech, it was Honbaza Petrkliciu, maybe you have seen it. In the Czech Republic, it was coordinated by Zuzana Musbergova from the Charles University of Prague. And for obvious language reasons, I was the coordinator of the project in North Macedonia, where I was born and raised. So we used voluntary participants that were counting the occurrence of different heterostylous forms in cowslips all across Europe. And I can proudly say that North Macedonia, being a teeny tiny country with only 2 million inhabitants, was in the top 10 contributors of Europe, not comparing to England and Estonia that have massive sampling efforts and massive communication efforts, but it was pretty good effort of contribution. My newest project, and if you want to talk about this more in detail after I'm done with the presentation, I'll be very glad to fill you in on more details, is parallel adaptation of plants to urban pollinators. So this is a project that is led by the University of Federico II in Naples, in Italy. And the original idea was to see how floral traits in Trifolium repens would evolve across an urban gradient where we know there is a change in pollinator composition and activity. Me and my current supervisor, being very creative and very innovative people, we decided that why just stay in southern Italy? We can also add climatic variation to this. So the new version of the project also involves the same type of studies replicated in a Central European climate, preferably the Czech Republic. So we are looking for help coordinators and some kind of support system in the Czech Republic. If you're interested, we can talk about it after the presentation. But my current pride and joy, and the project that I'm finally starting to have some substantial results with a lot of other things lined up, is the study of contemporary evolution as revealed by the resurrection method. So this project was led at the University of Ostrava, but also with the help of the Goethe University of Frankfurt and the University of Primorska in Slovenia, and was funded by the British Ecological Society, Johanna Quandt Young Academy, and Cost Action Conserve Plants. So now for the actual fun scientific part. You all know that the environment changes. This is not going to be a surprise to you. We have variation that are periodical, predictable, or oscillating, such as the changes of seasons. We have variations which are stochastic and randomly occurring, such as fires, floods, earthquakes. And we have changes that are directional and gradually go into a certain direction, such as the recolonization of habitats after an initial disturbance. Plants, being sedentary organisms, they have to find a way how to adapt to these environmental changes. And you know that this can happen through an evolutionary response, where there will be a shift in the allelic frequencies of the population in response to selective pressures. It can happen through phenotypic plasticity, where a certain genotype can produce different phenotypes in response to environmental variation. Or if the plants are capable and they're suitable habitats, they can migrate towards more suitable places when the environment becomes unfavorable. Obviously, all of these responses have their their limitations. Evolutionary responses depend on the amount of genetic variation, the adaptive potential present in the population. Phenotypic plasticity has physiological limitations because plants can only grow this big or this small, and migration is limited by the availability of suitable habitats. And today's plants face the added challenge of having to adapt to anthropogenic environmental modifications. You all know about climate change, about habitat fragmentation and degradation, and all kinds of modified species interactions, among which declining pollinators and the introduction of new invasive species on a regular basis. So plants can use this evolution, plastic, or migration response to adapt to it, and I'm specifically interested into what we call contemporary evolution, meaning an evolutionary change in allelic frequencies that occurs within a handful of generations. This is a study published by my PhD supervisor of the time, it's been a while, last year, where they studied the evolution of Viola arvensis natural populations in response to variation in the pollinator environment, where the populations from 20 years ago, they were exposed to a high pollination environment, and the pollinators gradually declined over the past 20 years. So there were a lot of traits measured related to plant attractivity to pollinators and the mating system. And it has been shown that in the descendants, the size of the corolla is smaller compared to the ancestors. In the descendants, nectar production is lower, and their overall attractiveness to bumblebees, their ability to retain interactions with bumblebees is generally decreased. On the other hand, the selfing rates, the self-pollination output, is actually increasing in the descendants compared to the ancestors. So within 20 generations, which is a teeny tiny amount of time from an evolutionary point of view, we have had a clear evolution towards increased self-pollination in response to pollinator decline. So this can, at least on the short term, help the populations to adapt to such drastic environmental changes. Otherwise said, contemporary evolution can rescue populations that are doomed to extinction. But how to study evolution? Even with contemporary evolution occurring over a few generations, this is a process that requires a lot of time, resources, and often both. Typically, what we can do is use the experimental evolution approach, where you take a starting population, you expose it to some kind of selective pressure, and then you wait for several generations. If you do this with microorganisms such as bacteria, it's fairly quickly because you have a generation time that spans a few minutes to a few hours. If you do it with plants, we're already talking about several years of experimental effort. Another approach would be the space for time substitution, where you compare the performance of two populations that have evolved into separate habitats, and you see if there are any differences that are potential adaptation to their respective environments. But this approach requires to rely on heavy assumptions about the population's evolutionary history and the common genetic ancestry. And obviously, no biological presentation these days is complete if we don't talk about genome sequencing, comparing the sequences of samples that have been taken in the past or in space can let us know if there are any allelic changes in the allelic frequencies, and whether they're adaptive or not, if we can have a functional analysis of these genes. Despite the plummeting costs of sequencing, this still remains a quite costly approach, especially when you are a young researcher starting without any substantial funds for this kind of efforts. So another approach would be to use this resurrection ecology, which is a very simple common garden experiment that kind of enables you to travel in time. At its very essence, resurrection ecology starts with a sampling of ancestral population at some point in the past, and the sampled seeds are preserved in suitable conservation conditions, usually in seed repositories, for several generations. then the exact same population is being resampled at present, and ancestral and descendant genotypes can be grown side by side in a common environment. So this is the basic resurrection approach. You compare descendants and ancestors in a common environment. If there is a shift in traits, you know that it is probably some kind of genetic differentiation. Whether it is adaptive or not, whether it is due to directional or random evolutionary processes, still remains the question. So you can combine the resurrection approach with other approaches, such as these semi-transplant assays, when you take the ancestor and descendant genotypes and you put them to the location of sampling. You can't put them in the past, but at least you can see if descendants are performing better than the ancestors at present. You can also try to simulate past and present day environments in growth chambers, for example, where you regulate temperature, moisture. You can try to regulate pollinator activity and pollinator abundance, and then compare in these reciprocal transplants in time the performance of ancestors and descendants. Obviously, you can couple the phenotypic analysis with genetic and epigenetic analysis to see to what kind of specific changes in the genome do the phenotypic shifts correspond. And resurrection ecology can be the endpoint of experimental evolution assays, where you take a starting population, you leave it evolve for several generations, and then you have preserved samples of your ancestral starting point and your descendant point to grow them side by side. So I use the resurrection ecology approach to study the plant adaptation to climate change and pollinator decline. Specifically, I'm interested into how do these huge changes in the environment that have occurred over the past 50 years affect the plant mating strategies and their floral traits. And this is important because floral traits, in their turn, by making the plants less or more attractive to pollinators, will affect plant pollination interactions, and modifying plant-pollinator interactions will have a feedback on plant mating strategies, and on the long term can further decouple or delay the pollinator decline. So specifically to how does pollinator decline affect the the evolution of flowering entomophilous plants, we have two mutually exclusive hypotheses. On one hand, plants can forego pollinator interactions, meaning that they will increase their ability to self-pollinate, so they don't need the pollinator services to reproduce. This is often accompanied with a decrease in the plant attractivity to pollinators, meaning that they produce fewer flowers, that are not as intensely colored, that they offer less rewards, and so on. This is what we call a selfing syndrome. And sometimes this can also be accompanied by an increase with the selfing ability, which can be also manifested as a physical decrease of a distance between anther and stigma, so that the pollen can easily reach the small distance between to its own stigma. The other possibility would be that in response to decreased pollination services, the plants would actually evolve to maintain their selfing rates, or rather to maintain their outcrossing rates. And for this, they would need to make themselves more attractive to the few remaining pollinators. This means that the plants should invest into producing more larger flowers that are more intensely colored, with more attractive rewards, hence they can be more intensely pollinated. And usually this should also manifest in the unchanged selfing ability, so no changes in hercogamy or configuration of the mating organs. So here is time for me to take a break, but I have a question for you. Which of these two strategies do you think would be more advantageous on the long term? Let me start the new question. And in case you have turned off the browser, you can still scan the same QR code or type in this link here. And I get 30 seconds break. Looks like nobody's trying to scan the code, so let's see. Very nice. it looks like when it comes to population, ecology, evolutionary potential, adaptive potential, you have all been very well trained here. Usually, it's not such a clear-cut answer that I get at this stage. But indeed, logically, we would think that maintaining pollinator interactions would be more advantageous, among other things, because of reduced risk of inbreeding depression, for example. So let's just look into this quickly, because it clearly shows that you already know what I'm going to talk about. So let's say that there is a decline of the pollinators in the environment, and the plants evolve towards increased self-pollination. This is accompanied by the reduced adaptive potential because of loss of genetic diversity through selfing, which eventually leads to an evolutionary dead end. Dead end never sounds good. On the other hand, the increased selfing can be accompanied by the selfing syndrome, which makes the flowers less attractive to the pollinators and further accentuates pollinator decline. So even though increasing self-pollination on the short term can reduce into overall increased number of offspring, the quality of the offspring reduces, and the interactions between plant and pollinators can end up being irreversibly decoupled. However, if the plants evolve to maintain pollinator interactions, that means that they should invest more resources into producing more and more attractive flowers. And this can be accompanied with less resources available to actually invest into their offspring, which can also lead to negative unwanted consequences and kind of limit the threshold of attractivity towards which the plants can evolve. So this is the background of what I do. And today I'm going to show you some quite preliminary results of 2 independent resurrection studies. And these results currently can help me answer 2 questions. Have trade shifts occurred in the last 30 years in natural populations of entomophilous plants? And are the observed shifts consistent with adaptation to pollinator decline and climate change? So to answer these questions, I relied on the resources of the European Native Seed Conservation Network, ENSCONET, which is a consortium that groups about 30 botanical repositories of seeds that anyone can freely access for research purposes. And with the help of experienced botanists, colleagues, we made a short list of about 25 species that are suitable for resurrection studies. The species were annual, which maximizes the number of generations between the ancestor and descendants. They were arable weeds, meaning these weeds that grow along the side of the fields, that are everywhere, easy to cultivate, fast cycling, but also important providers of ecosystem services, which we are currently losing with the current anthropogenic changes of the environment, especially in arable or agroecosystems. The species should be self-compatible, allowing for easy shifts of selfing and outcrossing rates. Entomophilous, because plant-pollinator interactions. They need to be sampled in a known location, so I can resample the descendants. And they should be sampled sometime between 1990 and 2010, because we know that the viability of stored seeds declines rapidly past 30 years of storage. And so among this short list of 25 species, last year, I was able to grow 2 species with somewhat contrasting ecological characteristics. A brassicaceae that is mainly distributed in the Mediterranean region, Matulatricus pedata, and a more an annual species with a more temperate distribution, Centorium erythrae. So I will show you the results of each of the species separately, and then I will try to bring them all together in the end. Let's start with Centorium erythrae. This is a species that is predominantly pollinated by bumblebees and hoverflies, but also by bees. It offers pollen as reward. Its mating system is obviously self-compatible and hercogamous, meaning that there is a reciprocity in the distance of anther and stigma in different configurations. And it flowers towards mid to late summer. This might be interesting information for you later on in the presentation. So from Centorium erythrae, I disposed of 2 populations, one sampled in Belgium in 1992 and then resampled in 2020, and another one sampled in Poland in 2003 and resampled in 2020. And I grew all of these plants in a common garden assay at the University of Ostrava in the teeny tiny but very nice botanical garden we have there. So before me showing you the results, I want you to stop and think, what would you expect? You have several different options about how the descendants should evolve. You can select multiple responses, and ideally you should select 3. One regarding the flowering, one regarding attractivity to pollinators, and one regarding their selfing ability. So I'm starting the question, and I get yet another break for myself. And this is actually 3 questions in one, so take your time to think about it, maybe pair up. I see more and more heads coming up, so I'm showing the results in 3, 2, 1. Okay. So general expectation is towards earlier flowering, towards increased attractivity to pollinators, and unchanged hercogamy. So this should be an adaptation to climate change, and these two should be an adaptation to maintain interactions with pollinators. Let's see now, what did I actually observe in my common garden experiment? So regarding the flowering phonology, the date at which the first flower was observed, unfortunately, I was busy running another experiment in Germany, so I completely missed the start of the flowering date in the ancestral Polish populations. Ancestors will always be in blue, and descendants will always be in red from this point onwards. But I did manage to see that the Polish descendant genotypes do tend to flower a little bit later in the season. And I was lucky enough that the Belgian genotype started to flower overall later, and the descendants seem to be flowering a little bit later than the ancestors, even though I don't have their full phenology. It's biology, it's how it works. I wasn't there, well, I was at the wrong place at the right time. But overall, even with the missing data, you can see that there are differences in the timing of flowering of ancestors and descendants, with the descendants flowering later than the ancestors. Now, this is usually contradictory to everything you have been taught about adaptation to rising temperatures in the spring, because the days get warmer earlier, so the plants should flower earlier to take advantage of these increased temperatures. But this is actually without counting the effects of summer drought. Centorium erythrae flowers in late July and August, which means that if it delays its flowering, it means that it avoids the probability of encountering intense summer droughts. So for late flowering annuals, delaying your flowering time in response to climate change might actually be an adaptive response. Regarding the attractivity to pollinators, I measured several traits. One of them was the height of the flowering stalk that bears the flowers, which kind of shows them off, waves them to the pollinators. And in the descendants of the Belgian population, we see that the flowering stalks are significantly taller than the ancestors, but no significant differences were observed in the Polish population. I measured the floral display, at least in the Belgian population, which is the number of flowers the population offers to the pollinators at a given point in time. And we see that in the Belgian populations, the descendants offer more flowers, they present more flowers to the pollinators than the ancestors. And lastly, I spent 2 painful weeks mounting and photographing and measuring flowers under a microscope at the University of Primorska, and I thank my colleagues there for being so patient and guiding me through the process, to estimate the shape and general parameters of flower structures. And regarding, for example, the petal surface, which is quite correlated with flower size, I demonstrate that in the Belgian population, as well as in the Polish population, the descendants have larger petal surface, so overall larger flowers than the ancestors. So in general, we see taller flower stalks that make the flowers more visible, larger floral displays that are more attractive to the pollinators, and larger petals, which are also more attractive to the pollinators, all of them being compatible with the idea that the descendants have evolved to have their flowers to be generally more attractive to the pollinators. And lastly, I checked about the configuration of the mating system, whether the anter-stigma distance has changed over the past 20 to 30 years. So that is a measure of the hercogamy in the plants. And I see no statistical differentiation between ancestors and descendants, although there is a somewhat higher variation in the Polish population here, and there is differences between populations in the degree of hercogamy. So we're now going to dive straight into Matthiola tricuspidata and those results to see how they compare between the two species. So Matthiola tricuspidata is a plant that is predominantly pollinated by bumblebees. It offers nectar as a reward, and its mating system is self-compatible even though it's from the Brassicaceae family. Its flowering time is in early spring. In southern France, the beginning of flowering is sometimes in March, maybe April if it's a particularly unfavorable year. I only had one population with a known location of Matthiola tricuspidata that was sampled in the south of France in 1997 and then resampled by colleagues in 2017. And I grew ancestral and descendant genotypes in this reciprocal transplant in time experiment where I simulated past environments and present-day environments by modifying the temperature, precipitation, moisture in growth chambers. So the question for you again, which results do you expect? This time you can choose at least two answers regarding the flowering timing and the attractivity to pollinators. There you go. Looks like people are debating even, which is a good sign for me. Should we switch to the answers? In 3, 2, 1. Okay. So we have earlier flowering leading for now, and again, increased attractivity to pollinators with people keep voting. Okay, so now let's see how this compares to the actual results that I had. Regarding flowering phenology, this was the experiment that kept me busy and made me miss the flowering phenology of Centaurum erythrae. So here I have very detailed counts of the first day of flower opening. And in both the ancestral environment, like the colder, wetter environment, and the descendant contemporary environment, which was warmer and drier, we see that the descendants seem to flower earlier, well, flower significantly earlier than the ancestors. And this is again compatible with the idea of adaptation to climate change for early spring flowering plants, where in response to increased temperature early in their development, they open their flowers earlier so they can take advantage of the pollinator abundance and so on. Regarding the floral display, the amount of flowers that the plant offers to the pollinators at a given time, again, in the ancestral and in the contemporary environment, we see that the descendants tend to offer overall more flowers to the pollinators at any given time, and this is a statistically significant difference. But when it comes to floral morphometry, I measured things like petal length and petal width, which did not differ between ancestors and descendants. However, I still don't have the full data set. These are results coming from the measurement of about 30 individuals out of the 200 for which I have preserved petals. I measured the angle of the petal, which turns to be somewhat smaller in the descendants than compared to the ancestors. Smaller, narrower angle means technically smaller petals, which is not compatible with increased attractivity, but again, very small sample sizes. Then I measured the length of the corolla tube, which can facilitate the access to the reward for the pollinators. I measured the size of the anthers, the size of the gynoecium, and none of these seem to show any statistically significant differences between ancestors and descendants. So when it comes to how did Mattheolatricus pedata evolve over the past 24 years, at least the population that we sampled in southern France, the descendants have larger floral display, maybe somewhat smaller flowers, and their reproductive traits have not changed. So overall, this is still kind of compatible with the idea that the population has evolved to maintain the interaction with the pollinators by making more, if not more, attractive flowers. So to sum it all up now, regarding the two species with contrasting ecologies, with contrasting geographic distribution, what have we learned so far? We see that there is trait differentiation between ancestors and descendants, which is consistent with evolutionary trait shifts because we have phenotypic differences between two temporal points in a common garden. Specifically, in Mattheolatricus pedata, the descendants flower earlier than the ancestors, whereas in the late summer flowering Centorium erythrae, the descendants flower later than the ancestors. And both of these, taking into account the reproductive ecology of the species, are compatible with the idea that flowering time has adapted to climate change. Then we also see that in Mattheolatricus pedata, the descendants have larger floral display and unchanged reproductive organ size, at least with my reduced sample. And the descendants have larger floral display, larger flowers, and again, unchanged hercogamy, unchanged size of their reproductive parts. So this is indeed compatible with the idea that these populations have evolved to maintain pollinator interactions despite documented decline of pollinators in their respective regions. So how do these results compare with other studies of contemporary evolution, whether it is through resurrection ecology or experimental evolution? In Adonis annua, this is again an annual bumblebee pollinated species, and resurrection studies showed that the descendants have overall large flowers, so they have evolved to maintain pollinator interactions. In Leontodin hispidus, a perennial Asteraceae plant, the descendants have evolved to have taller flowering stalks, making the flowers more visible, like waving from afar to the pollinators, again compatible with increased attractivity, maintaining the interactions with the pollinators through increased attractivity. But we saw at the beginning of the presentation that in Viola arvensis, the descendants have actually evolved to have smaller floral display. And another study showed that this is a study preceding the one from the beginning of the presentation. The descendants also have shorter leaf flowers and smaller flowers, so they invest less and less into maintaining the interactions with the pollinators. And finally, there was an experimental evolution study in Centorium erythrae in a semi-controlled environment, showing that in response to decline of pollinators, the descendants actually reduced hercogamy, reduced the distance between anther and stigma after only 5 generations of evolving with a lack of pollinators. So you can have support for both of the hypotheses, and it is highly likely that the evolutionary trajectory is not something that is convergent for all species and all populations, but it does depend on the local conditions to which the populations have been exposed. So where do I go from here? First of all, I'm currently finishing the cleaning of DDRAT SNPs data, which will help me to do FST, QST comparisons in order to disentangle between random evolutionary processes, such as bottlenecks, genetic drift migration, and selective evolutionary processes, such as selection for specific traits and differentiation in quantitative traits. The DDRAT SNP data will also help me to have direct estimates of selfing rates in natural populations of ancestors and descendants. For both of these populations, I have fruit and seed sets and very hardworking students that are currently counting the minuscule seeds of Centorium erythrae and Mattheolatricus pedata, which will help me to estimate autonomous pollination, because some of the plants were never exposed to pollinators. and also to estimate the fitness of the plants in open pollinations, which will help me to calculate selection gradients and see if there was directional, stabilizing, or divergent selection acting in the last 30 years in the populations. For Matiola tricuspidata, a very motivated PhD student, upon her own initiative, took measurements of pollinator behavior, especially the duration and the frequency of the visits of the pollinators. So soon we will know whether pollinators tend to spend more time harvesting rewards in the ancestors, which are seemingly more attractive compared to the descendants. So, with this data, I can say that at least in the populations that I have studied with the resurrection method, we do observe trait shifts that occurred in natural populations over the past 30 years, and they are generally compatible with the idea of evolutionary response to climate change and pollinator decline. But my work is far from done. I have another list of 23 species waiting to be examined that can help me further address questions, such as, are the observed trait shifts adaptive for the populations? Do they show any kind of convergent patterns between species? Can contemporary evolution provide evolutionary rescue? Because even if it increases temporarily the reproductive output, it doesn't mean that this increase is sufficient to compensate for demographic losses due to drastic changes in the habitat. And finally, the question that we would all like to be answered: will plant-pollinator interactions be maintained long-term? Like, this is a very tall order. I am not pretending that I can answer it by myself, but if you are interested into the resurrection approach and willing to implement an experiment like this with seeds that they have available, maybe we can discuss on how we can join our efforts to study this. So to finish off, I would like to advocate a little bit about the powers and limitations of the resurrection method. What is really great about this approach is that you have an almost instant demonstration of evolutionary trait shifts, because all you need is to grow ancestors and descendants together for one generation. Admittedly, you need to have appropriate samples from ancestors and descendants. The resurrection method can be easily upgraded to test the adaptive character of trait shifts by doing these reciprocal transplant in time experiments, by adding genomics, by adding experimental evolution, and so on. And it is a very nice way on how to value the genetic resources that we currently have in non-commercial seed banks. Botanical seed repositories are largely underfunded in Europe and across the world, and this is yet one more way of how to show that these resources are important, because not only they give us insight of what happened in the last 20 to 30 years, but maybe they can also provide gene resources for reintroducing more attractive traits into present-day populations. And finally, the resurrection sampling protocols are compatible with protocols about ex situ conservation of plants, which require the maximization of sample genetic diversity. Obviously, this is all very good, but resurrection ecology presents its own challenges. First one being the limited access to ancestral material. Not in the sense that seed banks are not willing to share the ancestral material, but that the sampling of these populations was not made with resurrection ecology in mind. So you get what you get. I started with a list of maybe 10,000 accessions, and I ended up with about 100, from which only 25 species, but maybe like 50, 60 accessions were appropriate for the resurrection study. Another problem is that even when you have properly conserved seeds that germinate well, you don't necessarily have data about how they were sampled in the past, because there was a time where naturalists would just go outside, pick up a few plants, be happy that they have some kind of sample from the population without taking care of whether it is representative of the distribution, genetic diversity, ecology of the species, and so on. Another thing is that for a lot of nicely preserved accessions that are still viable, we don't know where they came from. So we have great material, but we can't resample the descendants, unfortunately. And lastly, there is the problem of confounding effects of adaptive and non-adaptive evolutionary processes, because when you show a trait shift, it can be due to adaptation, to adaptive evolution towards maintaining interactions with pollinators, but it can be simply a random result because you only have one or a few populations per species. And the multi-species approach cannot solve this issue entirely, but it can at least show us if there are convergent patterns between populations with different ecological characteristics. So this was the part for the resurrection ecology. I just want to remind you that my resurrection project is one thing that I'm still working on, and I'm slowly implementing it as I get more and more fundings about it. But that right now I am establishing this new evolution of trifolium in response to urban gradients. And if this is something that you find interesting and potentially can be implemented at the university here in Brno, I am definitely looking for possibility to be hosted as a visiting researcher, to maybe collaborators. Well, I say here entomologists because this is really my weak side, but with Zdenka we already talked about possible other compatibilities in our skills. And were there any master students interested in doing field work, we can always find a way of how to have you somehow involved with the University of Naples and with the University of Brno here. And to finish off, I would like to thank you for participating actively by answering all of my questions. Give a big shout out to my friend Natalia Aguilera Lopez that did all the pretty pictures. Everything nice in the presentation is for her. Everything that didn't work, that's completely my fault. These are my coordinates here, so you can scan them if you want to get in touch later or just come and talk to me after this. Thank you again for listening.
Thank you, Bojana, for this talk. I'm really happy to have you here to see the presentation because it was really gorgeous. I enjoyed it. And I think it was also interesting for us because it was a little bit different point of view on the botanical research, and it's open new areas what can be interesting for us. So it's time for the questions. So if I will ask, if I will mix two approaches, the first one with the different population, and the second one you are starting in the urban area. The population can differ in the isolation.
Yes.
And I can imagine that population which are isolated will have species with high proportion of selfing individuals.
Potentially, yes.
Affect the results of your first study?
Absolutely, yes, absolutely. If this is also the issue, is that you cannot disentangle these demographic and habitat loss processes from the genetic processes unless if you know exactly what happened at different points in time. So this is all the struggle of why we are trying to combine the resurrection approach with marker data, because then we can at least see neutral and selective trait differentiation. But in the end, there is always the question of what we're observing here. Is it due to evolution? Is it due to drift? Is it due to adapted genotypes migrating from elsewhere? Or, and this is something that my preliminary marker data are hinting at, the original population went extinct, but we don't know it because we don't have like long-term survey data. And then it was recolonized by completely new genotypes. So these are questions that we cannot fully answer with the material that we have in European seed banks. But there is the project baseline, which is actually doing sampling of populations over time. They go and resample the same natural population every year to see how genetic frequencies change over the course of the year, writing down environmental parameters, the nearest by populations, and so on. So I think they're already in the 10th year of this kind of sampling. In the next few years, we can expect some very interesting results with this kind of data. But while waiting for this forward-in-time resurrection approach, where you start sampling now for the future, the best we have is European seed banks that have data that were sampled 20, 30, sometimes 50, 100 years ago.
Yeah. And the second question, thank you, is that probably the ancestor population could have some problem with the persistent seeds, I think. After 40 years, not all the seeds will...
Yes. Absolutely. So this is something that all resurrection studies are now being very careful about. First thing that we do is check the germination rates in a controlled environment. They were, in both of these cases, are above 80% for ancestors and descendants, which is quite high. then it's always the same thing. Like ideally, we would be able to genotype the germinated and non-germinated seeds and see if there are any differences in any kind of selection, any kind of differences in sensibility to heat between different genotypes, but again, too much money.
Yeah, I think that for this reason, these arable beets are great because they are annuals and they have this persistent seed bank. Usually they are forming that because they have to wait for some root crop systems and changes in the root crop, so it is good system for studying. But I think the species from different habitats will have different problems.
Absolutely, absolutely.
So I gave you some time, and now it's your turn. Milan.
Thank you, very, very interesting. I want to ask, when you measured the time of flowering and compared it between the ancestral and current populations, how did you measure it? Did you simply take the date of the first flowering plant and convert it, or did you use some more sophisticated statistics, like comparing the distribution of the number of flowering plants plotted against
Yeah.
the date?
So the data I have presented here is simply the date at which we observe the first fully open flower. But I do, well, at least in Matthiola tricuspidata, I have the detailed phenology of the population throughout the flowering period. So these are analyses that are going to come later.
Okay, so if there was a different result for Centaurium and Mattiola, could it be due to different methods used to assessing the shift in time period? Because to me, if only the date of the first flowering individual is counted, I would maybe think that it's not very, it may not be very robust estimation of flowering, because the pollinators maybe are not so much dependent on the first single flowering plant.
Mm-hmm.
They use the plant when the whole population is
Yes,
flowering.
absolutely. So from this point of view, the floral display at a given date is more representative of how attractive is the population to the pollinators, because yeah, having one flower that is exposed a week or 2 weeks or even 5 weeks early is not going to feed the whole pollinator population. But the flowering time is more a typical trait that is measured to see how plants respond to usually changes in temperature or the accumulation of degree days. And I don't try to connect it with attractivity to pollinators. It is more the floral display, the number of flowers at a given survey date, that would show if the population is more, could show that the population is more or less attractive to the pollinators.
I think it also has something to do with the interaction with pollinators, because okay, when the climate is warming, then many of the pollinators are active earlier in the season, and the question is whether the adaptation of pollinators is faster. I mean, phenological
Yeah.
changes are faster in pollinators in plants. Is there any literature on this? Whether there is a difference between insects and plants and the changes?
There probably is. I am more focused on the plant part, and the amazing PhD student that did pollinator behavior is looking into the pollinator part. One thing with respect to your comment about flowering phenology, one thing that I could do in Matthiola tricuspidata, for which I have the complete phenology, is to see the peak flowering date also, if that has changed between the ancestors and the descendants, because that's supposedly the time where the populations are the most attractive to pollinators. So I do have that data. I was just in a hurry to put everything nicely together for today. because initially I suggested that they come in May, where I would have the genetic data and everything, but well, time constraints wanted otherwise. So maybe if I'm back in May, we can talk about that with actual data to support it.
So what you showed were unpublished data, right?
Yeah.
Ah, I see. I was thinking it's already from the publication.
No, this is.
Yeah. So in that case, I would recommend just to make some robustness tests, like using different methods of measuring flowering phenology and see whether different methods give let's say consistent results.
Absolutely. Yes. Absolutely.
Okay, so you have this data about phenotype,
Mm-hmm.
and are you going also to check if there are some changes in genotype?
Yes. So for Centorium erythrae, I have this. I have almost all of the individuals sequenced for DDRAD markers because, well, working with non-model species, you don't have reference genome. It's always a problem. Not with Trifolium. So I have the DDRAD markers. Same thing. The data is here. I just need to take the time to start analyzing them. But yes, and for Mathiolatricus spidata, there were the original accessions were genotype, but they only have like 10 individuals from the ancestors and 10 individuals from the descendants genotypes, which can give us information about overall population genetic differentiation, again with DDRAD markers. But I can't do, for example, things like estimates of selfing rates or some more advanced quantitative genetic estimates.
Other questions?
And if you feel shy about asking questions in English, klidně se můžete ptát i v češtině, ale já budu odpovídat v angličtině.
Something that just depends on the availability of the genotype for that particular population, but in another region, the populations could go another way, or it's because for some species, some traits are more labile than for others, so the evolution follows lines of least resistance.
Yeah, that's a great question that I really want to answer, but with the limited availability of the samples is a bit complicated. So one thing that eventually, when I get the fundings to do extensive research, I'm planning to do is to try and sample populations contemporary populations only nearby the original locations and see if there are similarities in the traits, correlate them with environmental parameters and with genetic variation, and see if what we observe in our population is like a specific pattern to its genetic composition or to some kind of specific environmental trait, or if it is all the populations in the surroundings that share environmental conditions seem to be showing the same evolutionary trend. Like, this is, for now, the only way how I have thought of addressing this question. If you have any other ideas?
Regarding your planned study about Trifolium, why do you think that Trifolium is a good model for such studies? Because when I think about Trifolium, it has at least two disadvantages. One is that it's clonal. So one clone, one genotype can exist at a locality for many years, probably. And it will not change because it's one genotype. Which is a difference to your models, Centorium and
Exactly.
There is a good assumption that they will evolve relatively quickly. And the other is that Trifolium, I think, is used in seed mixtures that are sown in grasslands in the cities. So what you can find there
Absolutely.
So why did you select trifolium?
I mean, the short answer to that question is because I didn't write the research proposal. But if we look into the actual biological and implementation of the project, the issue with mixture with commercial seeds is obviously a question. And at least in southern Italy, we do have access on how the selected sites were managed to know that they haven't been sown with trifolium in the last 20 years. The other option would be, instead of working with trifolium, I always mix them. White clover is trifolium repens, yeah, and the red clover is trifolium pratense. So would be to work with trifolium pratense, which is not used in these commercial seed mixtures, but it is a bit less abundant, and to maybe try and compare how the two have changed also, depending on how much sampling effort I can put into the study. Regarding about the clonality, indeed, I've always privileged working with annual species because they're easy to cultivate, but also because from an evolutionary point of view, 20 years is 20 generations. But we do expect that at least 5 generations would have occurred in trifolium repens with overlapping between old clones and newly recruited seedlings. But this is just how things happen in nature. I'm not very good at expressing this argument, but yeah, when you study evolution of plants in response to urbanization, you need to take them as they occur with their life cycles and everything. We can't just try to force everything into models that are suitable for the studies. We also need to look into models that are a little bit more complicated, where we'll have to disentangle overlapping generation, shorter evolutionary periods in the amount of generation that have occurred, and so on. On the other hand, trifolium being clonal, it presents the advantage that we can take the exact same genotypes in the urban populations and then replicate them to put them into rural environments and urban environments and see what is the difference between them. So there are advantages, there are disadvantages. And the last thing about why trifolium was selected is that, well, it has been extensively studied, and it has been shown that 20 years is quite enough time to see a substantial changes in the floret size. Now, depending on in which direction do pollinator communities evolve, but obviously when pollination is more efficient, the floret size is the, what is the name of the top part of the petal? Well, anyways, so the petal size increases when pollinators are more abundant and when the plant is more efficiently pollinated within a reduced amount of generations. And so we already know that this kind of signal can be observed with a limited evolutionary time span, and we want to test it on a more extensive scale.
You started with the example of Lamium amplexicaulis.
Mm-hmm. And that was bound to happen.
But you presented the differences between proportion of autogamous flowers and entomogamous flowers. But I think this is not stable because these plants are also adapted to some seasonality. In the early spring, when the insects are not so active, so the proportion of cleistogamous flowers is bigger, I would expect. And I saw it in several weeds, like just also this Veronica, Persica, Polita, and so on, and also this Lamium. I think this could be somehow dependent on the seasonality. Maybe
I have also done studies about variation of claystogamy in response to seasonality, and indeed, in Lamium amplexicaulis, that is a spring annual, the spring plants produce more open flowers than the autumn plants. And it's most likely dependent to photoperiod and temperature. But my competition experiment was actually replicated in spring and in autumn to account for the seasonality factor. And, well, obviously there is no simple response because the final answer to my study is the effect of competition varies between populations, and it's also seasonal dependent. It's a complicated mess of interactions between factors.
And then the other one on the growing pond are open and
Exactly.
the season is later and there is possibly potential that they will be somehow cleaning.
Yeah, there is always the thing of, yeah, when you use an ecological approach, you cannot control for everything, and in my opinion, you should not try to control for everything, because that's just how it happens in the nature. So you will observe some variation. If your study is robust enough, the variation will not be clouding the overall interpretation, but maybe sometimes they're just not generalizable patterns.
Yeah, it's not kept in mind.
Yeah, well, you just mentioned it, that you would work with, you had genomics and epi in the in brackets, but then in the techniques, I don't know if anyone that you mentioned was actually incorporating methylation or some epigenetic pattern.
Good, yeah, another good question.
Do you measure that, or is it just a possibility that?
It is a possibility. It is a possibility that I wanted to implement this year, but now I will focus more on trifolium, because when you start talking about epigenetics, everybody's like, oh, but you're not working with clonal plants, and you're working with plants that do cross-pollination, so you have all this genetic variation that is going to be clouding your epigenetic variation. But one very simple approach developed by Vít Latzel from the Institute of Botany in Prague and in Třeboň, I think, is this demethylation technique that doesn't soak the seeds, which drastically decreases their viability, but just spraying the plants with a demethylation agent. And I know this is, again, this kind of ecological general approach with a lot of noise, but if we can show that demethylating ancestors and descendants decreases phenotypic differences between them, then we know that there is some kind of effect of epigenetic regulation that is worth exploring then with epigenome sequencing and so on. But that being expensive and more time consuming, we start with this simple common garden experiment, control of unmodified ancestors and descendants, and then demethylated treatment for ancestors and descendants.
So last chance for some questions. Otherwise, you have this code, so you can contact Bojana. So thank you for your presentation.
Thank you for the invitation again.